Genetic fingerprinting is a diagnostic tool used widely in forensic science, plant and animal breeding, and medical diagnosis. It's based on the fact that DNA of every individual, except identical twins, is unique. It relies on the fact that the genome of most eukaryotic organisms contains many repetitive, non-coding bases of DNA. 95% of human DNA is currently not known to code for any characteristic, but may yet be found to be functional. DNA bases which are non-coding are known as variable number tandem repeats, VINTers, VNTRs. For every individual, the number and length of VINTers has a unique pattern. They are different in all individuals except identical twins, and the probability of two individuals having identical sequences of these VINTers is extremely small. However, the more closely related two individuals are, the more similar the VINTers will be. Gel electrophoresis. This is used to separate DNA fragments according to their size. The DNA fragments are placed onto an agar gel and a voltage is applied across it. The resistance of the gel means that the larger the fragments, the more slowly they move. Therefore, over a fixed period, the smaller fragments move further than the larger ones. In this way, DNA fragments of different lengths are separated. If the DNA fragments are labelled, for example with radioactive DNA probes, their final positions in the gel can be determined by placing a sheet of X-ray film over the agar gel for several hours. The radioactivity from each DNA fragment exposes the film and shows the fragment is situated on the gel. Only DNA fragments up to around 500 bases long can be sequenced in this way. Larger genes and whole genomes must therefore be cut into smaller fragments by restriction endonucleases. The making of genetic fingerprint consists of 5 main stages. Extraction. Even the tiniest sample of animal tissue, such as a drop of blood or hair root, is enough to give a genetic fingerprint. Whatever the sample, the first stage is to extract the DNA by separating it from the rest of the cell. As the amount of DNA is usually small, its quantity can be increased using polymerase chain reaction. Digestion. The DNA is then cut into fragments using the same restriction endonucleases for all samples of DNA. Separation. The fragments of DNA are next separated according to size by gel electrophoresis under the influence of an electrical voltage. The gel is then immersed in alkali in order to separate the double strands into single strands. Hybridization. Radioactive or fluorescent DNA probes are now used to bind with the VINTers. The probes have base sequences which are complementary to the base sequences of the VINTers and bind to them under specific conditions such as temperature and pH. The process is carried out with different probes which bind to different target DNA sequences. And finally, development. An X-ray film is put over the nylon membrane with the fingerprint on it. The film is exposed by the radiation from the radioactive probes. If using fluorescent probes, the positions are located visually using UV light. Because these points correspond to the position of the DNA fragments that are separated during electrophoresis, a series of bars is revealed. The pattern of bands is unique for every individual except identical twins, hence the name genetic fingerprint. Interpreting the results. DNA fingerprints from 2 samples from blood found at the scene of a crime from a suspect are visually checked. If there appears to be a match, the patterns of bars at each fingerprint pass through an automated scanning machine which calculates the length of the DNA fragments from the bands. It does this using data obtained by measuring the distances travelled during electrophoresis. Finally, the odds are calculated of someone else having an identical fingerprint. The closer the match between the 2 patterns, the greater the probability that the 2 sets of DNA have come from the same person.